Hi. So today is gonna be interesting. We will discuss RNA sequencing and transcriptomic analysis. The main idea behind the video is that we're gonna be analyzing RNA sequencing data taken from a sample of peripheral mononuclear blood cells. Peripheral mononuclear blood cells include, for example, monocytes, T helper cells, cytotoxic T cells, and etc. So we'll be able to cluster these cells into group according to their gene expression RNA sequencing profile. The video or the lecture will be divided into two parts, technically two videos, starting with the theoretical section and then followed by the practical programming section. We will review programming in three different languages, including Python, MATLAB, and R programming language. So yeah, let's start. So now we'll start with the presentation. From the title, you can see Single-Cell RNA Sequencing Data Analysis, Applied Machine Learning Biological Model. Please note that this model is a simplified version of the real work that can take place. However, it shows the main idea of RNA sequencing and single-cell RNA sequencing. So we will start with the theoretical section, followed by the practical section. We have the objectives and the prior knowledge. Objectives: understanding gene expression. RNA sequencing as a measurement of gene expression. Methods of quantifying and measuring gene expression. Types of RNA sequencing: the traditional RNA sequencing vs the single-cell RNA sequencing. The main stages of RNA sequencing data analysis, including the normalization of the data, reducing the data dimensionality using algorithms like PCA or t-SNE, gene expression data clustering, such as using the k-means algorithm. The prior knowledge: be familiar with gene expression and nucleic acid. Probably you have taken this in your high school studies, maybe. Be familiar with basic genetic techniques. Basic genetic techniques, I mean PCR, adapters, or microarrays, and so on. Be familiar with different ways of data representation and visualization, like scatter graphs, heat maps, violin plots, and so on. Basic knowledge in linear algebra and statistics, descriptive and inferential. Will not dive deep into the statistics and linear algebra behind the algorithms that we'll use. However, you're expected to have a basic knowledge in these topics. So, what is gene expression, and what is the main idea behind gene expression? It's simply translating information stored within the DNA into gene products. So it could be simply defined as the production of mRNA and proteins from DNA during the processes of transcription and translation. You know the process. We have DNA in the nucleus. Gene expression takes place usually by transcription factors binding to the promoter regions of the DNA, triggering RNA polymerase activity, and leading to the formation of a transcription initiation complex. This leads to the production of a pre-mRNA. This pre-mRNA, after the spliceosomic activity and after certain modifications, it will be converted into mature mRNA with only exons and coding regions. This is exported into the ribosomes to be converted into proteins later during the process of translation. So determining the gene expression profile of cells is a highly important technique in biology, as the identity and the function of cells are linked to which genes are being differentially and actively expressed. So usually, we tend to measure the gene expression. We need to quantify gene expression during different circumstances or during different conditions. Gene expression is highly important, and changes in gene expression are also very important during processes like cell differentiation and specialization. So, for example, if we have a stem cell differentiating into an embryonic stem cell differentiating into, for example, a neuron, it will probably need a differential gene expression, changes in its gene expression profile. Most of the genes will be switched off. Some of the genes will be switched on that are required to its structure and function. The expression of particular genes can be measured by quantifying the levels of the gene's product. So the most obvious way to measure gene expression is by measuring the amount of the gene products. The gene products including RNA and proteins. This is the most obvious way. Although it is an indirect measure of gene expression, it is the easiest and probably the most effective way. So, what is RNA sequencing? RNA sequencing, it is one of the methods, genetic methods that are used to quantify and measure the extent of gene expression. So it's a method that determines the abundance of RNA transcribed from actively expressed genes in the genome, usually from a sample or population of cells. So RNA sequencing, we are indirectly having a measure of the expression of certain genes by quantifying the amount and the abundance of RNA transcribed from these specific genes. So how RNA sequencing takes place? RNA sequencing is a very complex process. Will not dive into the exact details of process. However, it involves a lot of steps and a lot of technical and practical details. So we will have a sample of interest. We will have the cells. By certain methods, we will extract the RNA from these cells. After extracting the RNA, we will carry out fragmentation. So we will break the RNA into smaller pieces. We will transcribe the RNA into DNA. So this is known as reverse transcription, converting RNA into DNA by using special enzymes known as reverse transcriptase or reverse transcriptases. These enzymes can convert mRNA into complementary DNA or cDNA, and usually the cDNA a cDNA library is created by adding certain adapter sequences to these cDNA fragments. After that, we want to sequence these cDNA molecules or these cDNA fragments, sequencing through processes like new generation sequencing, NGS. Simply, you can use a sequencing machine and a software which can give you the sequence of DNA in a special file format, which is called a FASTQ format. We'll not dive into the details of file formats, maybe for another video. After sequencing each cDNA molecule originating from RNA fragments that we have extracted from the sample of interest, we will carry out certain computerized methods to map the cDNA sequences into the reference genome or the organism genome. So if we are studying the human genome or we are extracting cells from human cells, we should map these sequences and align these sequences into the reference human genome. This is followed by the quantification and the data analysis. So after aligning the sequences into the reference genome, we will quantify each type of RNA. For example, as you can see here from the graph, this is the quantification bar graph. This gene has around 14 million read counts, RNA read counts. This gene probably has the lowest gene expression with around 7 million total reads. So the cDNA is sequenced by NGS or new generation sequencing, generating sequencing reads or simply reads that are mapped to the reference genome to identify their corresponding genes. The number of reads, the detected amount of cDNA from each gene, directly correlates with its mRNA level and thus its gene expression. So the total RNA or cDNA reads from a particular gene directly correlate with its gene expression or its value of gene expression. This gene has the highest gene expression. This gene has the lowest gene expression. So this is the main idea behind RNA sequencing. Now we have two types of RNA sequencing. We have the single-cell RNA sequencing and we have the traditional RNA sequencing. Usually, in the traditional way, we have a sample of cells and we extract the mRNA from the whole population. We are not specific to one kind of cell. No, we will extract the RNA from the whole population, which is known as bulk RNA input from the whole population, and we will analyze it. However, in single-cell RNA sequencing, we are able to isolate RNA and extract RNA from each individual cell, and we can analyze the RNA from each individual cell at the individual cell level. So it is a very precise and useful method. So what are the advantages of using such techniques, single-cell RNA sequencing? We will be able to measure the variability in gene expression among cells with similar identity or function. So for example, here we have a collection of tumor cells. These tumor cells, they might have similar identity, they might have a similar, for example, function. However, with single-cell RNA sequencing, we are able to differentiate between the cells that have minor differences. So for example, if we have a group of muscle cells, or we have a group of muscle cells or neurons of the same type, we could differentiate between these cells by looking out at minor differences in their gene expression. So this is one of the benefits of using single-cell RNA sequencing. Using single-cell RNA sequencing, we will be able to identify rare cell types in the population. Also, we will be able to characterize complex heterogeneous tissues. So as you can see here, we are able to produce and look at different type of cells. We can classify the cells according to their gene expression profile, and we can reveal any heterogeneity within the population, and we can create a subpopulation. This is a subpopulation. This is a subpopulation. This is a subpopulation. In bulk RNA analysis, we are just extracting RNA from the whole cell population. We are considering the whole cell population as one population of identical cells, and we are just measuring the average gene expression. So traditional cell population based RNA sequencing can determine the average gene expression profile of a whole cell population and not at the individual cell level. So yeah, this is a single cell RNA sequencing. RNA sequencing raw data analysis. Now, for the raw data analysis, we will describe this process in this video in a simplified way, and it's only for beginners. This might not be exactly the same as the real life bioinformatics way. However, it will be very useful for you to learn the basis of RNA sequencing data analysis. So what are the major stages? The major stages, we will start with normalizing the expression data. We will talk about this in details later. Log transforming the data, reducing the data dimensionality, and clustering gene expression patterns, which is the main topic of this video. We will be able to cluster the cells and create subgroups of cells depending on their gene expression profiles. We can represent RNA sequencing data in a wide variety of graphs and a wide variety of data representation depending on what we want to see exactly. Different ways of visualizing data can lead to different conclusions. We can produce different conclusions from different ways of visualizing data. Most commonly, we use heat maps, scatter plots for clustering, and more. Other types of data visualization include Violin plots, box plots, and many more, volcano plots. And so now we will talk about the first step of data analysis, or RNA sequencing data analysis. How to normalize the data, or before raw data format and normalization. So usually RNA sequencing data are displayed, the final RNA sequencing data are displayed in a form of a matrix, which could be a text file, a MATLAB file or matrix file, or even a CSV file. Now, you should understand that from the process or from the step of extracting the RNA into producing this table, there are multiple steps that need to be carried out in order to produce this final table or counts table. And this table is then considered as the raw data for the next step, which is the data analysis step. So if we return to here, we have a series of steps taking place after the mapping to create this table. And this table, the raw expression values, is considered to be the start point of the data analysis and quantification. So usually the raw numbers represent the quantity of RNA reads. So for example, here we have cell 1, we have cell 2, we have the expression values, RNA sequencing expression values of gene 1 and gene 2. For example, here we have 10 RNA reads of gene 1 within cell 2, 30 RNA reads of gene 2 within cell 1, and so on. Now, an important step to be carried out on these expression reads is normalization. Normalization is extremely important. It accounts for differences in the total number of reads, RNA extracting efficiency per cell. So, you might say here that gene 1 in cell 1 has higher expression than in cell 2. However, after normalizing the data, you will discover that gene 1 has equal expression in both cells because during the normalization, we are taking into account the total number of reads within each cell. Here in cell 1, we have 40 total number of reads. In cell 2, we have only 4 total number of reads. So you could imagine that if we have extracted 40 total RNA reads from cell 2, then we will probably have a value of 10 here instead of 1. However, due to different RNA extraction efficiencies per cell, we are not having equal figures. At the same time, we are having equal expression. And this is shown by the normalization. The expression of gene 1 in cells 1 and 2 is the same when taking into account the total number of reads from cell 1 and cell 2. So normalization is extremely important during this step. Normalization is also important for other reasons. How we can normalize the data. Usually we will have an RPKM calculation. RPKM calculation is a normalization process of making measurements comparable across the cells. So how we can measure the RPKM value? Usually the gene expression values are shown as RPKM values. This measure normalizes the reads per kilobase per million reads. So the RPKM is equal to 10 to the power of 9 times C over N times L. Now what is C? C, it's simply the number of mappable reads on a feature, transcript, or exon. So to C, it is the number of RNA reads or cDNA reads that have been mapped onto the reference genome and onto a specific gene, like gene 1. So for example, for gene 1, cell 1, we have a value of C equal to 12. The length L, it is the length of that feature in kilobase. So we should take into account during normalization the length of each gene in kilobases. Why we should take into account the length of each gene? Because genes with greater lengths, genes that are longer, are more likely to have more reads aligned to them during mapping. So let's return to this. If we have a very long gene, then it's more probable that we will collect more RNA fragments or cDNA fragments. So our next step in our data analysis, after normalizing the data, we should transform the cell normalized data. So usually initial normalized data will follow a skewed distribution due to the presence of extreme values like 0. If the data is not close to follow a normal distribution, then there will be violations of certain assumptions that we'll use later. So if we return to this table, usually in our RNA-seq data, normalized RNA-seq data, we will observe a lot of genes with a 0 expression value. And you can see here genes with a normalized value of 0 may not be expressed, or they might be from cells with relatively few total reads. So usually we have a lot of expression values showing as 0, and this will correspond with this skewed distribution. As you can see here, we have a skew near to 0. So how we can reduce this skew and make the distribution closer to being normal? Why we should have an data to be nearly normal distributed? This is due to the statistical tests and the parametric tests that we will use later during our data analysis that might require data with that follow a normal distribution. So we can use a certain transforming methods like log transformations to get rid of extreme values and reduce the skew. How we can log transform the value? We can log transform the value by adding 1 to each value. So we will have non-zero values. All 0 values will become 1, and any other values also will be incremented by 1. And then we will take the natural log or the log base 2 of the positive values that are produced. So in this way, we are reducing the skew and making the data look like being normal distributed, more like being normal distributed. So what are the benefits of transforming the cell normalized data? Reducing the skew and representing log fold changes. So also we are able to represent changes at the logarithmic level. So if the changes are very high relatively, we will be able to visualize these changes easily by using this logarithmic plot. So now we're going to be discussing reducing dimensionality. So usually we have RNA sequencing data, transformed cell normalized data. They will be in a tabular format. They will be in a table. As you can see here, we have a multi-dimensional table with several columns and several rows. So how we are gonna be visualizing this data? Probably we should reduce the dimensionality of this data. As humans, we are only able to visualize a maximum of 3 dimensions. If we are studying the expression of, for example, 2000 genes, then it will be impossible to visualize 2000 dimensional space. How you can plot the expression value for a single cell across 2000 genes. You can't visualize a point in space with 2000 different coordinates. This is impossible. As humans, we use the convention of X, Y, and Z coordinates representing the 3 dimensions that we are able to visualize. Above that, we will not be able to visualize such data. So we can use special algorithms to reduce the dimensionality of our data while preserving most of the variance. Algorithms exist to reduce the dimensionality of data. The most simple one is known as the principal component analysis. Principal component analysis is based on calculating eigenvalues and eigenvectors. It is a linear algebraic method used, and it's quite efficient in preserving the variance of the values. So for example, if we want to reduce 5D data into 2D data, we will lose some of the variance on our data. We will not be able to capture 100% of the variance of our data. However, we will preserve most of the variance using algorithms like PCA. Other algorithms exist like t-SNE, t-distributed stochastic neighbor embedding. This algorithm mainly depends on t-distribution, and we will discuss it later. It depends on t-distribution. It's more complex than PCA, and it has some certain advantages over PCA. Other algorithms exist, like the UMAP algorithm. The UMAP algorithm is relatively very complex. Uh, it depends on certain bases in algebraic topology and Raymond's geometry. Today we'll discuss PCA and t-SNE mainly. So the first algorithm, PCA, Principal Component Analysis. In PCA, you want to find a line along which most of the variation in the data is captured. The first best fit line is PC1, or Principal Component 1. So as you can see here, we have three-dimensional data, and luckily we are able to visualize such data. We will draw the first line, which is the PC1 or Principal Component 1. It is basically the line of best fit through the points. PC1 can be calculated using eigenvalues and eigenvector. There are specific methods. You can look out for these methods using the internet. This method tries to find the best way to rotate the axis of our data, such that the first axis captures most variation in the data, followed by the second one, and so on. So then we will draw another line, which is a Principal Component 2 or PC2. PC2 will be perpendicular and orthogonal to PC1. So now, if we take PC1 and PC2 as our relative x and y axis, we will see that we have reduced the dimensionality of our data from 3 into 2 dimensional data, 3D reduced into 2D. Although we have not preserved 100% of the variation in our data, at least we have preserved maybe 8% of the variation, and we can calculate it using specific computational methods. So suppose we have, for example, a 3000 dimensional data. We can summarize the 3000 dimensional data into, for example, 50 or 80 principal components. So you could imagine, if I draw here, you could imagine drawing PC3 and then PC4 and so on. If we have 3000 dimensional data, we can, for example, summarize the data into 50 or 80 principal components while preserving most of the variation. Usually, PC1, the first principal component, will preserve the most variation, followed by PC2, PC3, PC4, and so on. Maybe reaching PC100, PC100 maybe will contribute to 0.0005 of the variance of our data. So PC1 is the most significant axis, followed by PC2 and so on. So this is the main principle behind PCA algorithm. Now let's have a quick review over the t-SNE, t-distributed stochastic neighbor embedding algorithm. So you should know that it's a more advanced method for reducing dimensionality in a non-linear way. So usually we can divide dimension reduction algorithms into linear algorithms and non-linear algorithms. In the PCA, we are basically drawing straight lines, and it is a linear method. It's a completely linear method, depending on the basis of linear algebra. However, when compared to t-SNE, t-SNE is a non-linear dimension reduction way. So as you can see here, we have a parabolic curve, while for PCA we are having a straight line. It is a statistical method for visualizing highly dimensional data by giving each data a location in 2 or 3 dimensional map. So it is a non-linear dimensionality reduction technique well suited for embedding high dimensional data for visualization in low dimensional space of 2 or 3 dimensions. So it's a usual dimension reduction techniques. It depends on constructing certain probability distributions, statistical probability distributions, using the points in some way. The presentation will be put in the description. You can check the presentation and check the links to know more about how t-SNE works. The advantage of t-SNE over PCA is that t-SNE is better at preserving the local and spatial relationships between the points. So as you can see here, PCA, if you compare, here we are having data in a two-dimensional space, and we are reducing data in two-dimensional space into a one-dimensional space. As you can see here, the spatial relationships between the points is not preserved. So you can see an overlap between the black and the orange points. However, t-SNE, with its nonlinear nature, it will be able to preserve a smooth spatial relationship between the points. So if we reduce 2D data into 1D data, you will see that the relationships were preserved, spatial relationships were preserved. So this is the main advantage of t-SNE, and I think this is the best visualization to know the difference. Although this is not the exact mathematical interpretation, however, it's a good way to understand it in this way and visualize it in this way. So now, after we have reduced the dimensionality of our data, now we should cluster the data. After reducing the dimensionality of our data, now it's time to cluster the data into groups. Each cell will be classified into a group depending on its RNA-Seq gene expression data. With the practical section, we'll be working on peripheral mononuclear blood cells. As I said earlier, we have various types of cells. So we'll be able to classify each cell into a group. So this cluster, for example, will represent T helper cell. This cluster will represent cytotoxic T cell. This cluster, for example, will represent macrophages and so on, depending on the RNA-Seq gene expression profile. Clustering algorithms can be used, like the k-means algorithm. So as you can see here, before the k-means, we have just data reduced into two-dimensional or three-dimensional space. After performing the k-means, we'll be able to know or classify data into clusters or groups. So what are the inputs into the k-means algorithm? We have unlabeled data. Initially, we will have unlabeled data, so we'll provide the k-means algorithm with the coordinates or the points. For example, you can imagine here the y-axis being the PC2 and the x-axis being the PC1, and we can provide the k-means algorithm PC1 and PC2, or for example, we can provide the k-means algorithm PC1, PC2, PC3, PC4, and so on. The algorithm will be able to classify using a certain mathematical technique each cell into a specific identity or specific cluster. We should input into the k-means algorithm the number of clusters or the number of different types of cells. So for example, if we have 6 different types of peripheral mononuclear blood cells, we should tell the algorithm that we are having 6 different cells or clusters. Here, for example, we are having a k value of 3. So the algorithm will be able to divide the points into 3 groups or clusters. Sometimes we're not sure how many types of cells we have or how many groups we have. So we tend to plot an elbow plot to know the number of groups or the number of clusters that should be input into the k-means algorithm. So what is this method? Basically, we will try the k-means algorithm. We will try the k-means algorithm with different values of k. For example, here we are trying with values 1 to 10, and we will calculate the variance, the total sum of the variance. The elbow point or the inflection point, as you can see here, the x value here 4. This means that we should input into the k-means algorithm a k value of 4, and probably we are having 4 different clusters of data. This is the elbow point. What will be the output of the k-means algorithm? For example, here we have the cell number, cell 1, cell 2, cell 3, cell 4, cell 5. The output of the k-means algorithm will be a simple data structure like this, for example, a table. And for example, cell 1 is classified into cluster 2, cell 2 is classified into cluster 3, cell 3 is of the same identity as cell 2, it's classified into cluster 2, and so on. So this is the main idea behind the k-means algorithm and clustering.